Welcome back to this next video in which we are discussing influenza. So we're currently in the process of doing a bit of background, and we're discussing the different types of epithelial lining of the respiratory tract, and we've now been over one of the really important types, which is so important that it's actually called the respiratory epithelium, because so much of the respiratory tract is covered by this type of epithelium. Another name for it is to call it a ciliated pseudostratified columnar epithelium, and this nicely describes its properties. So we have seen that this epithelium consists of three major types of cells. The main type of cell is the ciliated columnar cells, which are columnar cells, meaning that they're very tall. Their height is approximately 4 times their width. They are ciliated, so at their apical surface they have these finger-like projections, and something that I actually missed out in the final video is to talk about what the cilia are actually going to do and which direction the mucus is going to be wafted in. We'll come to that in just a moment. So you have these ciliated columnar epithelial cells, which are the major type of cells of this epithelium. Then, dotted around the place, you're also going to have these goblet cells, which are also columnar, but they're not ciliated. Now, remember, I told you about mucus production by the respiratory epithelium, and I told you that it's a common misconception that the goblet cells produce all the mucus. That's not true. The ciliated columnar cells produce a huge amount of the mucus. Mucus has a huge number of different components in it. They produce a lot of the components and make a very viscous secretion. Goblet cells secrete a really important component of mucus called the mucins, which they add into the mucus that the ciliated columnar cells have produced, and these make the mucus more viscous. So overall, both of them work together to create the mucus. The goblet cells secrete a specific component of the mucus, which is the mucins, which increase the viscosity. Okay, but they do not by any means make the mucus full stop on their own. Then the third type of cell is the basal cells, which are the stem cells. They are the cells from which all the other cell types stem. They are the cells which are responsible for producing the two other cell types, the ciliated columnar cells and the goblet cells. And remember, the way in which they do this is they can divide to produce two daughter cells. One of the daughter cells will remain a basal cell so that you maintain the cell population of basal cells, whilst the other will go through a differentiation process and can become either of the two major types of cell that populate this epithelium, so the ciliated columnar cell or the goblet cell. Okay, and remember, the basal cells are much less tall than the other types of cell, and therefore they're squashed at the bottom like this, and that gives the epithelium the appearance of being pseudo-stratified. It appears as though it's got multiple different layers, even though actually it's just a single layer of cells. All the cells are attached to the basement membrane. Okay, so that's the respiratory epithelium. We talked about which portions of the the respiratory tract that are actually lined by this type of epithelium. We saw that the nasal cavity, the nasopharynx, the bottom portion of the larynx, the trachea, and the airways are all lined by this. Now let me come on to the function of the mucus and the cilia. So we'll start with the bottom portion of the larynx, the trachea, and the airways, and I'm sure you've heard this described before. So, all of these portions are lined by this epithelium. The tubes are all lined by this type of epithelium. Now, that means that on the surface of all of these tubes there is a layer of mucus, and the function of this mucus is that if any dirt particles or pathogens, so bacteria, viruses, parasites, fungal cells, if any of these come in, if they're breathed in with the air, they will hopefully get stuck in the mucus of one of these airways, or the trachea or the bottom portion of the larynx. They'll get stuck in the mucus lining one of these tubes, and then what gradually happens is the cilia waft the mucus upwards and outwards. So this is the direction that the cilia will be moving when we're talking about the bronchi and the trachea and the bottom portion of the larynx. They'll be moving to move the mucus upwards and outwards, and then the idea is the mucus is gradually brought up and then it's swallowed down the esophagus and it goes into the stomach, where of course it will burn and burn and burn. And this is what is called the mucociliary escalator. So I'll just put this down here. So the function of this mucus is, it's a key component, of course, of the mucociliary ciliary escalator, which is involving the mucus in the cilia, and it's escalating the mucus upwards and outwards, and anything that's trapped in that mucus will therefore be cleared from the respiratory tract, and this is absolutely brilliant. It is an absolutely brilliant way of keeping the lungs clean and free of infection. It really, really is brilliant. This is the reason that you do not all the time get respiratory tract infections. Without this, and indeed this gets broken in horrible diseases like cystic fibrosis, and they get really, really ill. They have respiratory tract infections continuously. Without this, you do get respiratory tract infections continuously. It's really, really important for keeping the lungs nice and clean. Now, you also have the same sort of thing in the nasal cavity. So you have cilia in the nasal cavity and also the nasopharynx, and these will move the mucus backwards so that it can then again be swallowed and go down the esophagus again to the stomach, and it can burn and burn and burn. But of course you can also get mucus coming forwards, and then it can be removed from the nostrils. Okay, so you also have this ciliary action to move mucus in the nasal cavity as well, and again it's the same principle. It's to try and catch dirt particles or pathogens when they're coming in the inspired air. This is just earlier on than this portion down here. Okay, so there's the function of the mucus and the cilia that I didn't quite get around to explaining in the previous video. So, next type of epithelium. So remember we had two more types of epithelium to go over. The next one that I'm going to do is the non-keratinized squamous stratified epithelium of the oral cavity, the bottom portion of the nasopharynx, the oropharynx, the laryngopharynx, the esophagus, and the top portion of the larynx. Now, this is a very, very tough epithelium, far tougher than this type of epithelium. This is a single layer of cells, this epithelium. That means it's not particularly tough. We're going to have a much, much thicker epithelium lining these cavities here, and the reason is that these cavities are exposed to food particles. Food is going to be coming in here quite regularly, and it's going to be rubbing up against the epithelial linings, and therefore the epithelial linings need to be quite tough, which is why we have this different type of epithelial lining here. Now, importantly for us, this epithelial lining is not susceptible to influenza infection, so that is why when you get flu, you do not get problems with your mouth. You do not get horrible mouth pain because the virus has infected your mouth, the epithelium of the mouth. It doesn't do that. It doesn't infect that epithelium because it's a different type of epithelium that's not susceptible to the viral infection. So let's now, need the moving part. Let's now discuss this type of epithelium. So we'll go over here. So this has a great big long description as well, so I'll begin by writing it all out. So this is going to be a non-keratinized, which, well, what does this mean? This means that the cells are not going to become keratin overloaded. So this is a squamous stratified epithelium, and there are two major types of squamous stratified epithelium, keratinized and non-keratinized. Keratinized is the type that the epidermis is, that lines the skin, and that is incredibly tough. Non-keratinized is slightly less tough, but I'll come back to what this word means later on and describe it better once we've got a bit more under our belt. So non-keratinized, squamous. Now, squamous is like columnar. It describes cells. Squamous cells are very different from columnar cells. Columnar cells were big, tall cells, very, very tall, whereas squamous cells are extremely flat. So this epithelium is going to have some squamous cells, some very flat cells. Specifically, the outer layer of cells are going to be extremely flat. So non-keratinized, squamous, stratified. Stratified we've already been over. That means that it's going to be multiple layered. It's going to have multiple different layers of cells, and then epithelium. Okay, so this is the type of epithelium we now want to discuss, non-keratinized, squamous, stratified epithelium. Now, just a little point early on. We're going to have multiple layers of cells because it's a stratified epithelium. So, are all of the cells in all the different layers going to be squamous? The answer is no. It's only the outer layer of cells that is going to be squamous. So when you have a stratified epithelium, you describe the type of cell by the outer layer of cells. So you have lots of different layers of cells, and they all have different shapes. The type that you use in this description is the type of the outermost layer of cells, and the outermost layer of cells in this type of epithelium are going to be squamous. So that's why we have squamous here. So this is a multi-layered epithelium, and the way in which we name the different layers of a squamous stratified epithelium is extremely complicated. There are loads of different naming systems. I like an extremely simple one, and that's the one that I'm going to share with you today. But if you go onto the internet, you will be able to find loads of other different ways of naming up the different layers of squamous stratified epithelial cells. I'm not going to use those. So that's just a warning. We're going to use a nice simple one. So let me start again by putting in a basement membrane. So all epithelia have a basement membrane which all the cells are sitting on top of. So again, we'll start here, and because we're going to have multiple layers of cells, we're going to need to leave quite a lot more space. So I won't label it up because we know what this is. This is a basement membrane. This is a meshwork of proteins that is very, very rigid, and which the bottommost layer of cells are going to be attached onto. Okay, so let's then start with the bottommost layer of cells, and this is called the basal cell layer. Now, this, just like the basal cells in the epithelium that we've just discussed, the respiratory epithelium, these are responsible for creating all of the other cells. And in contrast to in the respiratory epithelium, where the basal cells aren't usually active, they only need to become active if the epithelium gets damaged and you lose some cells. In the squamous stratified epithelium, you are continuously having to renew the epithelium. The outermost layer of cells is continuously being shed off, and you're continuously having to produce new cells. So the basal cells are going to be active all the time in a squamous stratified epithelium. So let me show these. So here we go. And I've said this already, but I'll say it again. This type of epithelium is very similar to the type of epithelium that lines the skin. The epidermis of the skin is a squamous stratified epithelium. The difference is that it will be a keratinized squamous stratified epithelium. But you know, I'm sure, that the epidermis is continuously being renewed, skin cells are continuously being shed off. It's exactly the same here. This squamous stratified epithelium of the oral cavity, the oropharynx, the bottom portion of the nasopharynx, etc., it's continuously being renewed. Cells are continuously being shed off. So here are the basal cells, and they're just like the shapes of the basal cells that we saw in the other epithelium. They're this sort of cuboidal shape is the way you describe them. They're not as tall as columnar cells, but they're certainly not flat cells either. So I'll finish this up. We'll have 8 basal cells in our picture here. So these are all basal cells. So this layer would be called the basal layer. Okay, so these are all stem cells. They're the cells that will produce all of the cells of the layers above them. And again, they will divide to produce 2 daughter cells, and one of the daughter cells will remain a basal cell so that we can maintain the basal cell population, and the other one will then go through a differentiation process and will change, and it will move off the basement membrane and it will move upwards. So to make this absolutely crystal clear, if this basal cell chose to divide, and they're all continuously dividing, when it divides, it will produce 2 cells. One of them will remain where it is, so it will remain in this position and will maintain the basal cell population. The other one will move into the layer above this. Now, the basal layer of cells is a single cell thick. So there's a single layer of basal cells. So this layer is a single layer of cells thick, whereas the other 2 layers that I'm going to have in my classification are going to be multiple layers of cells thick. So as I said, I'm going to pick a nice simple classification, and in my nice simple classification there are 3 separate layers of the squamous stratified epithelia. So the next layer, let's go up to the next layer now. So I'll pick some interesting colour. We'll have purple. So the next layer is what's called the midzone of the squamous stratified epithelium. And this contains cells that are going through the differentiation process. This contains the daughter cells from these basal cells that have moved upwards. Now, they are on their way to becoming squamous cells, to becoming these flat cells that will be on the outer layer of the epithelium. And before they become flat, they become bigger. So they become bigger and blobbier from these cells that are in the basal layer. The basal cells are beautiful, nicely cuboidal. The midzone cells are going to get bigger and more blobby. So let me try and draw this. So you're going to have multiple different layers of cells here. So I'll draw the topmost boundary of the midzone here. And now I'll just draw loads of cells. So here is a big blob of a cell. Here's another big blob of a cell, another midzone cell, and they should all be roughly the same size and same sort of shape. So here we go. And I'll overall get 3 layers in here. In reality, there will be far more layers than 3. It will maybe be up to about 10 layers of cells on top of one another, but I don't want to have to draw 10 separate layers, so we'll have 3 in my picture. So I think that gets the message across. So these are the progeny of the basal cells in the basal cell layer. So they move upwards and they start the differentiation process and they gradually become bigger and bigger. Now, once they have begun this differentiation process, they will not divide again. So the only cells that are dividing in this squamous stratified epithelium are these cells in the basal layer. These cells in the layers above are not dividing, they are just differentiating. They're changing their characteristics, and in the midzone layer, they become big and blobby, and they gradually move up the midzone. Okay, so let's now go to the layer above the midzone. So in green here, we then have the superficial zone. And again, the superficial zone consists of multiple layers of cells, not a single layer of cells, unlike the basal layer. Now, in the superficial zone, the cells have become flat by this point. So let me draw these on here. So again, we'll have the uppermost boundary here, and let's just draw a few cells here. So they've become flat. So here's 1, here's 2, and how many layers should I put in here? I think we'll actually make 5 separate layers here. And they still have their nuclei like so. So I might draw another section of them here. So again, you have lots of separate layers of cells in the superficial zone. And overall, the non-keratinized squamous stratified epithelium is estimated to have 20 layers of cells in total. A lot of them will be in the midzone there. So the superficial zone is maybe a little, in my picture, maybe it's a little bit of an underestimate to just have 6 layers, but we didn't have too many more, maybe 7, 8, 9 layers of cells, etc. So, in the superficial zone then we have these squamous cells, these flat cells, and these are these cells on the midzone that have differentiated further. So the progeny of the basal cells, they undergo this differentiation process, they make their way up the midzone. In the midzone they have this appearance where they've become big and blobby, and then by the time they get up to the superficial zone they become incredibly flat. And then they'll make their way up to the topmost layer of the superficial zone, and then they will be shed. So they'll be shed off. So I'll just move a little bit. So the most superficial layer of cells in the superficial zone will be shed off, and then there will be a new layer of cells underneath that will replace it. So the whole thing is continuously being regenerated. Cells are continuously moving up and being shed off. So the basal cells have got a lot of work to do. They have to continuously be dividing to produce new cells that are going to replace the ones that are being shed off. And that, I'm afraid, is the life cycle of a squamous stratified epithelium. The cells do not live long. They are continuously being shed off. They go through this process only to be shed off. Okay, so that's the structure of a non-keratinized squamous stratified epithelium. Let me now talk a little bit more about this non-keratinized. In fact, I'll talk about all the words in this title again. So, there are lots of different epithelia in the body that are squamous stratified epithelia like this. And this picture would apply very well to all of them. You can always divide them into these three layers: the basal cell layer, the mid-zone, and then the superficial zone. The squamous stratified epithelia of the body can be divided into two different types: non-keratinized and then keratinized. The example that I have drawn here is non-keratinized because the cells still have their nuclei in the outermost layer. In a keratinized example, and the major example of a keratinized squamous stratified epithelium is the epidermis of the skin. In a keratinized example, what happens is as the cells undergo the differentiation process here, they become absolutely overloaded with this protein keratin. Keratin is a cytoskeletal component, so remember, inside cells you have a huge number of fibres that are holding the cell in its shape. That's known as the cytoskeleton. Keratin is a protein that is a component of the cytoskeleton. So, in a keratinized squamous stratified epithelium, the epidermis of the skin is the major example. The cells, as they undergo this differentiation process, become absolutely full with this cytoskeletal protein keratin, and they become so full of keratin that they actually lose their nuclei by the time that they are in this outermost layer of cells of the superficial zone. So the outermost layer of cells in the epidermis, the keratinized squamous stratified epithelium, is just, they have no nuclei, they're just full of keratin, they're just bags of keratin. And this is extremely strong, and this is important in the skin where we need absolute rigidity. We need, it needs to be very, very strong. So that's why you have keratin overloading in the epidermis of the skin. In the oropharynx, the oral cavity, the nasopharynx, the bottom portion of the nasopharynx, the laryngopharynx, and the top portion of the larynx and the esophagus, you don't need quite the same level of protection as the skin needs. So these are non-keratinized squamous stratified epithelia. And there are other examples of non-keratinized squamous stratified epithelia in the body. For instance, the anus is lined by a non-keratinized squamous stratified epithelium. A lot of the genital mucosas are lined by this as well. So in the female, the vagina and the vulval mucosa, and also the ectocervix is lined by non-keratinized squamous stratified epithelium. In the male, the preputial mucosa that lines the inside of the foreskin is a non-keratinized squamous stratified epithelium, and in both male and female, the urethral lining is a non-keratinized squamous stratified epithelium. So there are multiple examples in the body of non-keratinized squamous stratified epithelium, and in these, the cells do not become keratin overloaded in the same way as they do in the epidermis, and they maintain their nuclei, so the outer layer of cells still have nuclei. They are not just bags of keratin. Okay, so that's what the non-keratinized means. Squamous refers to the fact that the outer layer of cells is squamous. Stratified, hopefully that's obvious, there are multiple layers of cells. All of these other layers of cells are not in contact with the basement membrane, so this truly is a stratified epithelium. Okay, so this is a non-keratinized squamous stratified epithelium. I hope that you've got some key messages here about this type of epithelium, and one of the key messages is that it's continuously renewed. It's continuously having the outer layer of cells shedding off, and you're creating new cells that are replacing that, so it's continuously being renewed in this way. The basal cells are continuously dividing in order to achieve this continual renewal. Okay, so let's just then finish off by talking about, reminding you of which portions of the respiratory tract are lined by this type of epithelium. So it's all the portions that might come into contact with food. The oral cavity is lined by this, the bottom portion of the nasopharynx is lined by this, the oropharynx is lined by this, the laryngopharynx is lined by this, the top portion of the larynx is lined by this, and of course the esophagus is lined by this. All of these portions are lined by this type of epithelium. And this type of epithelium is not susceptible to influenza virus infection. So all of these parts are not actually going to become infected with influenza virus, which is why I wanted to explain this histology to you, and hopefully you can kind of realise why this epithelium is so unsusceptible to influenza. The fact that it's continuously being renewed in this way means that, you know, the virus is going to be in contact with the outermost layer of cells, but they're not going to live that long at all. They're being shed off continuously. So how is it ever going to sort of establish itself when the cells that it might actually go into are just going to instantly be shed off and are going to die? So that's why influenza doesn't establish itself in the squamous stratified epithelia, because it can't get into the important cells, which are the basal cells, because they're right at the bottom. Whereas in the respiratory epithelium, these ciliated columnar cells, they're much longer lived. They stay there for a long, long time until something causes them to die, and then they're replaced. So these are the ones that are actually going to be infected by influenza, which is why influenza affects the nose. The portion of the throat that it infects is the nasopharynx. That's why you get a sore throat. It does affect part of the pharynx, but it doesn't affect these other portions of the pharynx. It'll be able to infect the bottom portion of the larynx and the trachea and even the bronchi. And as we'll see, horribly, it can infect the alveoli. Hopefully it doesn't. In most people, it never gets to that point where it infects the alveoli. That's when you've got viral pneumonia, primary viral pneumonia, but it can in principle. So, the next thing we now need to go on to is the epithelium then of these alveoli, because these, as I say, are also susceptible to influenza virus infection. So let's just briefly go over the structure of the alveoli then. So we'll do this here. Okay, so let's do the structure of alveoli. So we'll start again with a basement membrane. The alveoli, these little air sacs, they are still lined by an epithelium, and that epithelium will be sat on a basement membrane. So let's just draw this, and we'll draw two alveoli. So here is one alveolus, and here is another alveolus. So this is the basement membrane. Okay, and the epithelium of the alveoli is a simple epithelium, i.e. it's a single layer of cells. And there are two types of cell of the epithelium. There are very, very flat cells, so there are squamous cells, and then there are cuboidal cells, and these are called type 1 and type 2 pneumocytes respectively. So let me just show this now. So I'll get my blue pen back. So I'll firstly show you some type 1 pneumocytes, because these are the major type of cell of the alveolar epithelium. So here's a type 1 pneumocyte, and you can see that these are clearly supposed to be extremely flat. So there's another type 1 pneumocyte, here's another one, and it continues on. Now let me put in a type 2 pneumocyte, which is cuboidal, like so. So here is type 2 pneumocyte, then we'll have another type 1 pneumocyte, and most of the surface area of alveoli are coated by type 1 pneumocytes, which are these squamous ones, like so. But I'll put in another type 2 pneumocyte here, and then we'll have another type 1 pneumocyte here. Okay, so that finishes the epithelium here. So, doing the description that we've done for all the other epithelia, we would describe most of the cells here as being squamous, and it is definitely a simple epithelium. Simple means that it's a single layer of cells. All of the cells are attached to the basement membrane, and it does not appear as though it's multiple layered epithelium. So it's not pseudostratified in the way the respiratory epithelium is. Okay, so the complicating factor about the alveolar epithelium is that we have two different types of epithelial cells which are given special names. So the major type, the squamous type, are the type 1 pneumocytes. So pneumo is a prefix which means pertaining to the lungs, and then cyte means, of course, cell. So the type 1 pneumocytes are the squamous ones, and of course the purpose of these type 1 pneumocytes is just to be extremely thin, because remember the function of the alveoli. Here is the air, so this is the air sac, this is the actual alveolar sac, this is where the air is, this is where fresh air is. And of course, we want gas exchange to occur between the air in the alveolar space and the pulmonary capillaries, which are underneath the basement membrane. So let me just put in a little pulmonary capillary. I'll do it here in blue. So this can represent a little pulmonary capillary here. And you should know that underneath the basement membrane there'll be connective tissue with the pulmonary capillaries running in them. So here is a pulmonary capillary, and of course blood will be flowing through here. You'll have deoxygenated blood coming in here with a high carbon dioxide in it, and then the idea of course is that oxygen will come in, so from the air, the fresh air, you'll get oxygen coming in, and from the blood you'll get carbon dioxide coming out, so you'll hopefully get gas exchange, and then the oxygenated decarbon dioxided blood will then go off and back to the heart and be circulated around the body. So that's why we have these extremely thin alveolar epithelial cells, so that we can have good gas exchange, because we don't want to have a thick diffusion pathway for the gas to get through, because then we'd get less diffusion. So that's the purpose of the type 1 pneumocytes, to have a very thin barrier so that gas exchange is maximized. The other type of cell is the type 2 pneumocytes, and if you remember your respiratory physiology, you might be able to tell me, or tell your computer screen, what the function of the type 2 pneumocytes is. These are the cells which produce surfactant, which is extremely important. It's a molecule that is secreted onto the surface of the alveolar epithelium, and it's really important for decreasing water tension or surface tension that would otherwise cause the alveolar air spaces to collapse down. So these are tiny little spaces, and because there is fluid on the surface of these alveolar epithelial cells, there is the temptation for the alveolar space to collapse in on itself. So remember, a good way to describe surface tension, a good analogy to use, and in fact a perfectly correct analogy to use, is when you were doing histology and you have two of those little glass plates on which you put the specimen. If you put two of the glass plates on top of one another, so let's say here's one glass plate, and then I'll do another glass plate in green here, so here's another glass plate. If you put them on top of one another and those glass plates are perfectly dry, then it is really easy to then pull the two glass plates apart. There is nothing holding the two glass plates together. If the glass plates were perfectly dry and you put them one on top of the other, like I've shown in this picture, it's really easy to then pull them apart. This is common knowledge. If you then were to wet the surface of these glass plates, so you were to stick both of the glass plates in water so that their surfaces are now wet, and you were then to stick them together, you would have a much more difficult time pulling them apart. And that is because there is a surface tension force now that the surface of the glass plates is wet. It's exactly the same with the alveolar epithelial lining. When this is wet and this is wet, if they were to come together, it would then be very difficult for them to break apart. You'd have to put a lot of force into it. That is called surface tension that is produced there. The surfactant that is produced by the type 2 pneumocytes is there to prevent surface tension. It's a special molecule that prevents this force from being established. So indeed, if you were to dip these wet glass plates in surfactant, it would then be very easy to pull them apart. The surface tension would be destroyed, and it's exactly the same that's true in the alveoli. So type 2 pneumocytes, they produce surfactant. This is just a bit of background knowledge. This isn't terribly relevant to us. Okay, so you have these two types of epithelial cells lining the alveolar spaces. Type 1 pneumocytes, which are just a squamous cell, and their function is just to make the diffusion distance as small as possible between the alveolar airspace and the pulmonary capillaries. And then you have the type 2 pneumocytes, which have a very different function. They're cuboidal epithelial cells, and their function is to secrete surfactant, which is involved in stopping surface tension forces from causing the alveoli to collapse. And of course, if all the alveoli were to collapse in your lung because of surface tension, then the lung would be made completely useless. So surfactant is extremely important. Okay, so this is the lining then of the alveoli. I've shown two separate alveolar spaces here, and these are the epithelial cells that line them. The major type being these type 1 pneumocytes, which are these squamous cells. Okay, these cells are susceptible to influenza infections, so both the type 1 and type 2 pneumocytes can be infected by influenza virus. As we'll discuss when we come on to the pathogenesis, this does not normally happen in most people. Thankfully, the virus does not usually get right down into the alveolar spaces. If it does get right down into the alveolar spaces, then you are extremely ill and you have what we call primary influenza viral pneumonia. Okay, so I just want to finish my discussion of the histology then of these epithelia by talking about the fact that underneath the basement membranes, what you will have in the case of all of these epithelia, so in the case of the respiratory epithelium, the non-keratinized squamous stratified epithelium, and the alveolar epithelium, and I should actually just put that as the title here, this final epithelium, we will be calling that from now on the alveolar epithelium. Underneath the basement membrane, you're going to have connective tissue, and in that connective tissue, you're going to have immune cells, and this will be important when I want to come on to discuss the inflammatory response to having an influenza infection. So underneath here, you're going to have immune cells. And so sentinel cells, cells that are standing guard in your tissues, waiting for signs of infection, and if they find signs of infection, they kickstart the inflammatory response. So let me just write down the name of these cells then. So I'm just now discussing the fact that you have sentinel cells underneath all of these epithelia. So sentinel cells are present in all tissues in the body. Examples, the two key examples are dendritic cells and macrophages, and their job is to stand guard. Their job is to look continuously for signs of infection or signs of danger, signs that cells in the local tissue that they're present in are being damaged by some horrible thing. And if they find signals like that, then they call for help. They call for the inflammatory response. They call for troops to come into that tissue that will help to eradicate whatever it is that is causing this problem. And all of the troops are in the blood, and therefore we need to get them out of the blood. And that process by which you get these troops to fight infection out of the blood and into a tissue is called the inflammatory response. So these sentinel cells are involved in kick-starting the inflammatory response, and the two major types are dendritic cells and macrophages. So all over the body, not just in the epithelial linings of the respiratory tract, but all over the body, whatever tissue you go to in the body, you have these sentinel cells that are standing guard, looking for infection, and if they find signs that there is infection, they kickstart the inflammatory response, they call for help, and we'll discuss in much more detail later on the nature of the inflammatory response, how these cells call for help, they release special molecules, but we'll come on to that much later. For now, all I want to tell you is that they are there, and I want to add them into the picture. So let me add in on this picture of the alveolar epithelium some macrophages and dendritic cells. So let me start by putting in a dendritic cell then. So we'll go for this yellow colour for a dendritic cell. So dendritic cells, the reason they are called dendritic cells is that they have dendrites, which are these processes coming off the main cell body. Now, they have absolutely no relation whatsoever to nerve cells. Nerve cells, of course, have dendrites as well, but the word dendrite just refers to the process coming off the cell body. So let me just draw one of these dendritic cells. Where shall I put it? I'll put him in here. So here he is. He's got these sort of like tentacle-like processes, these dendrites as they're called, coming off like so. And here is the nucleus of this dendritic cell. So that's a dendritic cell, and you can see that one of these dendrites is going through the gap between two of the type 1 pneumocytes. It goes through the basement membrane, through the gap between the type 1 pneumocytes, and therefore part of this cell is exposed to the alveolar space, and this means that the dendrite can be looking for signs of pathogens in that alveolar space. And this is why dendritic cells have dendrites. It's so that they can extend into loads of different portions of the tissue looking for signs of pathogens. It's a brilliant design. If you were to design a sentinel cell, this is probably what you'd come up with, having these long tentacles, dendrites I should call them rather than tentacles, extending into the surrounding tissue looking for signs of pathogens. Okay, and these even extend into the alveolar spaces by going between the type 1 pneumocytes, like so. Okay, so there is a dendritic cell. Now let me put in a macrophage. Now, macrophages are going to be in two places in the alveolar epithelium. Of course, you will have macrophages in the underlying connective tissue. So here is a macrophage. And by the way, I should specifically call it a resident macrophage. Now, as part of the inflammatory response, when it occurs, you bring in a huge number of macrophages from the blood. Of course, they don't circulate as macrophages, they're monocytes in the blood, but when they come out of the blood, they differentiate into macrophages. However, in all the tissues of your body, you have resident macrophages, macrophages that are permanently there, performing the role of sentinel cells, guard cells, looking for signs of pathogen invasion. And this is what I'm drawing now. So remember, macrophages are big blobs of cells, so here is a big blob of a cell that's a macrophage here. Now, you'll have macrophages not only in the connective tissue underlying the basement membrane, but you actually also have them in the alveolar spaces in the alveoli. So here is one of these alveolar macrophages, as they're called. So this is an alveolar macrophage, and I'm just going to introduce you to a little symbol way of writing macrophage. So a shorthand way of writing macrophage is to put M and then the Greek letter phi, like so. So this is an alveolar macrophage. So that's an abbreviation that I quite like to use for macrophages. And people even do use that in clinical scenarios. So in the clinical notes, you'll see M5 for not macrophages necessarily, but monocytes usually, because that's what we measure in the blood, but they are the precursors to macrophages. And dendritic cells, an abbreviation that I'll use for dendritic cells is DCs. Okay, so here are the sentinel cells added into my picture of the alveolar epithelium then. In the alveolar spaces you have these alveolar macrophages that are looking for signs of pathogens, and of course both of these types of sentinel cell, they are also phagocytes. They're capable, if they find signs of pathogen, not only of initiating help signals for the inflammatory response, but also of actually phagocytosing that pathogen, engulfing that pathogen and trying to break it down. But we'll talk about all of that much later on. Okay, and you then underneath the basement membrane you have the dendritic cells and macrophages in this connective tissue where the pulmonary capillaries are present in the lung parenchyma. Underneath the other two types of epithelium you'll also have these types of cells. So I'm not too bothered about this non-keratinized squamous stratified epithelium because of course this isn't going to be infected in influenza infection, therefore we're not going to actually be discussing inflammation occurring under here, but certainly in the connective tissue underneath here you will have resident dendritic cells and resident macrophages that are there as sentinel cells to look for signs of pathogens, and if they find them they will set off the inflammatory response and also phagocytose them. But I am interested in adding them onto this picture of the respiratory epithelium because of course the respiratory epithelium is going to be infected by influenza virus, and therefore it is going to be important that we know about these cells when we want to come on to discuss the inflammatory response occurring underneath these epithelia. Okay, so let me add them in here. So let me put a dendritic cell in. So again, the dendritic cells will have dendrites that extend through the basement membrane and in between the epithelial cells, like so. So this one is a dendritic cell, and you can see that I've drawn a dendrite there extending between two of the epithelial cells. Well, imagine there's an epithelial cell here, so that it can look for signs of pathogens actually on the epithelium. And then of course we'll also have resident macrophages, so I'll add one of these in here in the connective tissue underlying the basement membrane. Now, unlike in the alveoli where you had alveolar macrophages actually in the lumen of the alveoli, you don't have macrophages in the lumen of, for instance, the nasal cavity or the trachea or the bronchi, so they're a feature only of the alveoli, but you definitely do have them in the connective tissue underlying the epithelium. Okay, so relief. You can sigh a great sigh of relief. We have finished the histology part of the video, and we have nearly finished the background portion of the video. We will soon be going on to discuss the actual influenza viruses. The final thing that I want to do in this portion of the background knowledge is discuss the different types of respiratory tract infections. So let's just put a title here and do that. So, for short, respiratory tract infections are often abbreviated down to RTIs, standing, of course, for respiratory tract infections. And this is any infection of the respiratory tract. So any bit that we've looked at here, an infection of the nasal cavity, the pharynx here, the larynx, the trachea, the airways, or the alveoli, all of those are called respiratory tract infections, RTIs. Now, respiratory tract infections are often divided into upper respiratory tract infections and lower respiratory tract infections, and for short, upper respiratory tract infections are abbreviated to URTIs, and lower respiratory tract infections are abbreviated to LRTIs. So, what is the difference between an upper and a lower respiratory tract infection? Where is the boundary between what you would call an upper respiratory tract infection and a lower respiratory tract infection? Well, the boundary is the glottis. We use this as the boundary between an upper respiratory tract infection and a lower respiratory tract infection. So if you have an infection of the nasal cavity, that is above the glottis, so you would call that an upper respiratory tract infection. If you have an infection of the nasopharynx, that is above the glottis, and you would therefore call it an upper respiratory tract infection, and so on. Any of these portions here that are above the glottis, you would refer to those as upper respiratory tract infections. An infection below the glottis will be called a lower respiratory tract infection. So an infection of the larynx would be called a lower respiratory tract infection. An infection of the trachea would be called a lower respiratory tract infection. An infection of the airways of the lung would certainly be called a lower respiratory tract infection. And an infection of the lung parenchyma, the alveoli, is most definitely a lower respiratory tract infection. So it is the glottis that marks the difference, the boundary between what you would call an upper respiratory tract infection and what you would call a lower respiratory tract infection. Now, in influenza, generally you get both an upper respiratory tract infection and a lower respiratory tract infection. Influenza viruses generally cause infection of the nasal cavity, the top portion of the nasopharynx, which are both lined by respiratory epithelium. It won't infect the oral cavity or the bottom portion of the nasopharynx or the oropharynx or the laryngopharynx or the top portion of the larynx, and it certainly doesn't infect the esophagus because that's the wrong type of epithelium as we now understand. But it also generally infects the larynx, the trachea, and the start of the airways usually. And therefore, influenza is both an upper respiratory tract infection and a lower respiratory tract infection usually. Hopefully, as I said earlier, it doesn't usually get all the way down to the alveoli. If it does, you are very, very ill. So, some final pieces of terminology then. There are words for inflammation occurring in all of these different areas along the respiratory tract, and I just want to go over those now. So, we'll put them under the headings of upper respiratory tract infection and lower respiratory tract infection. So, when you get inflammation occurring in the nasal cavity, and understand, I'm talking about inflammation, not infection. These words mean inflammation in an area, not infection. Now, of course, usually when you get infection in a certain area, you do get inflammation. So often the terms are used, you can use the term really to mean infection of that area, but strictly speaking, these terms mean inflammation in the area that is usually as a result of infection in the area. So there is a term for infection of the nasal cavity, and that is, sorry, not infection, I've just made the exact mistake that I was warning you against. Inflammation of the nasal cavity, which is rhinitis. So rhinitis means inflammation in the nasal cavity. Working our way back, when you get inflammation of the pharynx, any portion of the pharynx, and of course in influenza we're going to get infection of this top portion of the nasopharynx and therefore inflammation in that top portion of the nasopharynx, that is called pharyngitis. So pharyngitis is inflammation of the pharynx. Okay, then working our way downwards to the larynx, laryngitis is the name for inflammation of the larynx, and this will be a lower respiratory tract infection now. So, inflammation of the larynx is laryngitis. Inflammation of the trachea is called tracheitis. So all of these things are going to occur in influenza because we're going to get infection of the nasal cavity, infection of the top portion of the nasopharynx, infection of the bottom portion of the larynx, infection of the trachea, and we'll get inflammation in all of those areas, so we'll get all of these words here. Then coming down to the airways, inflammation in the airways is called bronchitis. Strictly speaking, bronchitis means inflammation in the bronchi, as is obvious from the name. However, we use bronchitis to mean inflammation in all of the airways, even the tiny little airways called bronchioles. There is another word that you can use. You can use bronchiolitis, but you will never hear anyone actually use the term bronchiolitis. Bronchitis is what we use to mean inflammation in the airways generally. And then if the inflammation is in the actual lung parenchyma, so right down in the alveoli, there is a totally different word now used. All of the others have this nice suffix itis, which is what is usually used to mean inflammation in that area, and suddenly we now use pneumonia for inflammation in the alveoli, in the alveolar spaces, i.e. in the actual lung parenchyma. So pneumonia is inflammation of the lung parenchyma, inflammation in the alveoli as a result of infection usually. And in fact, that's actually a key difference. There is another word, the word that you'd have expected, which is pneumonitis. So we obviously know that that means pertaining to the lungs. Pneumonitis is what we would have thought would be inflammation of the lung parenchyma. Now, pneumonitis does mean inflammation of the lung parenchyma, but when the inflammation is due to a pathogen, an infective cause, you do not use the term pneumonitis. Instead, you use the word pneumonia. So pneumonia means inflammation of the lung parenchyma as a result of an infection, whereas pneumonitis means inflammation in the lung parenchyma as a result of something that isn't an infection. So when you get an autoimmune inflammation in the lungs, which you can do in certain horrible diseases, that would be referred to as pneumonitis rather than pneumonia. So this is just, again, a piece of background, a side piece of knowledge. We are obviously not going to get pneumonitis in influenza. If you do get inflammation in the lungs, in lung parenchyma in influenza, it's going to be of infective origin, of course, and therefore it will be called pneumonia. But just for your background knowledge, pneumonitis refers to inflammation in the lung parenchyma as a result of autoimmune causes. Okay, so that now completes the background knowledge. We have talked about these pieces of terminology for inflammation in certain areas of the respiratory tract. We've talked about the fact that respiratory tract infections can be split up into upper respiratory tract infections and lower respiratory tract infections, and the boundary between the two is the glottis of the larynx. We've also then talked about all of these pieces of terminology for inflammation in these different areas. In influenza, we're going to get infection of the nasal cavity and the top portion of the nasopharynx, and therefore we're going to get rhinitis and pharyngitis when inflammation follows the infection. We're also going to get infection of the larynx, the bottom portion of the larynx, and the trachea, and the early portions of the airways, usually in an influenza infection. Therefore, we're going to get laryngitis, tracheitis, and bronchitis when inflammation occurs because of that infection. If you are very, very, very unlucky, what can happen is the virus can get further and further along the airways to smaller and smaller airways, and then eventually might actually get down to the alveoli and therefore cause inflammation in the actual lung parenchyma, and then you have pneumonia. Then I gave you this other term, which is pneumonitis, which again means inflammation of the lung parenchyma, but not due to infection, due to autoimmune causes, usually, or some other inflammatory disease that isn't an autoimmune disease, but which isn't responding to infection. Okay, so we'll have a break here. In the next video then, we will actually start the bulk of the video. We will discuss the influenza viruses.